Good evening. My name is Crystal Gail Hammack, and I am going to do my presentation on cell cycle regulation. I do want to go ahead and let everybody know that I will have a video at the beginning and a video at the end showing myself. But when I go over my slides, I don't like to have my video in the corner because I have found that it's a distraction because it's something that's moving and it's not an actual video that I want them focusing on versus the actual content and just listening. So thank you for joining me today on cell cycle regulation. In this presentation, we will explore the importance of cell cycle regulation, focusing on how it maintains cellular health and prevents diseases, particularly cancer. Cell division is essential for growth, repair, and normal cellular function, but it must be tightly regulated to ensure that cells only divide when they are healthy and conditions are optimal, because we don't want to make bad cells over and over. We want to make healthy cells so we can be healthy. Without effective regulation, uncontrolled cell division can occur, which will cause cancer and/or tumors. The objectives that we will cover today are defining the cell cycle and its phases, describing key regulatory proteins such as cyclins and CDKs, explaining the importance of checkpoints, and discussing implications of dysregulation. The cell cycle starts with the G1 phase, which stands for gap 1. This phase is critical because it's when the cell undergoes significant growth in size and prepares for the rest of the cycle. During G1, the cell is not only increasing in physical size, but also in the production of essential proteins and organelles, which, remember, the organelles are like the little organs of the cells. These resources are necessary for DNA synthesis, which happens in the next phase. This phase is also when cells perform most of its normal everyday functions, depending on its type. For example, liver cells would be actively detoxifying harmful substances, and muscle cells would be working to maintain muscle tone and repair. Importantly, G1 includes checkpoint mechanisms that monitor whether the cell has grown enough and has the necessary resources to proceed to its next phase. If anything goes wrong, like DNA damage, the cell can pause in G1 or enter a resting state known as G0 until it's ready to continue. After the G1 phase, the cell enters the S phase, which stands for synthesis. This phase is focused on DNA replication. Each chromosome is carefully duplicated during the S phase to ensure that the resulting daughter cells have identical genetic material. This is essential for the stability of genetic information across generations of cells. DNA replication is a complex process that requires precise regulation to prevent errors. Specialized enzymes, such as DNA polymerase, play a critical role here by assembling the new DNA strands using the original strands as templates. A notable checkpoint mechanism also operates here to ensure that replication proceeds correctly without errors. If DNA damage or replication issues occur, the cycle can pause to allow the DNA to repair, or, if necessary, cell death to prevent the spread of mutations. Following the S phase, the cell enters the G2 phase, or gap 2. This is the final preparation phase before mitosis. In G2, the cell continues to grow and produce proteins, particularly those necessary for mitosis, such as microtubules, which are structural components that will help separate chromosomes during cell division. The G2 phase also includes another important checkpoint to verify that DNA replication was successful and that there are no errors or damage. This checkpoint ensures that the cell's genome is stable and ready for division. If the cell successfully passes this checkpoint, it's cleared to proceed to the M phase for mitosis. If not, the cell might not pause here to repair any issue, or if irreparable, trigger apoptosis or programmed death cell. And I'm sorry, I said may not. It will pause there, and check that also. Finally, we reach the M phase, which includes mitosis and cytokinesis. Mitosis is the division of the nucleus and has several stages: prophase, metaphase, anaphase, and telophase. During these stages, replicated chromosomes are aligned, separated, and distributed to opposite ends of the cell, ensuring that each daughter cell receives an identical set of chromosomes. After mitosis, the cell undergoes cytokinesis. This process divides the cytoplasm, completing the formation of two daughter cells that are identical to the original cell. Together, mitosis and cytokinesis are essential for growth, development, and tissue repair in multicellular organisms. After cytokinesis, each daughter cell enters the G1 phase, restarting the cell cycle. So it's going to do the whole thing all over again each time it makes a new cell. And if you look over here, I have an example of the cell cycle. So here's growth in normal metabolic roles, which is the G1, and then the S phase, which is also interphase. This is DNA replication. And then right here we have G2 for growth and preparation for division. And then right here, the mitosis, which includes prophase, metaphase, anaphase, telophase, and cytokinesis. Right over here, you see the G0, and it pauses, and then it just basically does the little loop, and it starts again. The cell cycle is regulated by complex mechanisms that control the progression through its various phases, including growth, DNA replication, and division. These regulatory mechanisms play a critical role in ensuring that cells divide at the right time, maintain the integrity of their DNA, and produce healthy daughter cells. One primary function of cell cycle control is to ensure that DNA is accurately replicated and that any damaged DNA is repaired before the cell progresses to the next phase. This control process involves checkpoints that pause the cell if errors are detected, allowing the cell to correct issues before continuing. This is crucial for maintaining genetic stability and preventing mutations from passing on to new cells. Uncontrolled cell division is a hallmark of cancer. When regulatory mechanisms fail, cells may begin to divide uncontrollably, leading to the formation of tumors. Proteins like cyclins and cyclin-dependent kinases, which are also known as CDKs, are essential in regulating these. Checkpoints with tumor suppressors like p53 playing a key role in halting the cycle if there is damage to the DNA. And this is a video that came from YouTube, and it's about the cyclins and the CDKs for cell cycle regulation, just to help bring it all together. Sorry, it won't let me skip the ad, so we're just going to have to go through the two ads.
expression factor. When the cyclin D, CDK4, and cyclin D, CDK6 complexes partially phosphorylate RB, it loosens its grip on E2F. E2F can then activate transcription of the cyclin E gene. Cyclin E binds CDK2, and this complex fully phosphorylates RB, completing its inactivation. Cyclin E also phosphorylates P27-HIF1, an inhibitor of cyclin D. Phosphorylation of P27-HIF1 taxes the degradation. Degradation of this protein promotes expression of cyclin A. E2F also promotes transcription of cyclin A by removing the repressive molecule cell cycle responsive element, or CCRE, from the promoter, allowing the cell to enter S phase. The action of E2F on cyclin A expression is part of a negative feedback loop, since the cyclin A CDK2 complex phosphorylates E2F, preventing it from removing the repressive molecule. Cyclin A is the only cyclin that plays a role in the regulation of two cell cycle stages. It can activate two CDKs, CDK2 and CDK1. When cyclin A forms a complex with CDK2, it allows the cell to progress to S phase. Meanwhile, binding of cyclin A to CDK1 allows entry into N phase. During S phase, cyclin A resides in the nucleus. It directs the initiation and completion of DNA replication. Critically, cyclin A ensures that DNA replication only occurs once per cell cycle by preventing assembly of excessive replication complexes. How does it accomplish this? Well, remember how cyclin E is around at the end of G1? One way in which it helps the cell move into S phase is by initiating assembly of the pre-replication complex. This complex is needed for DNA replication to occur and forms at the origin of replication. However, cyclin A also associates with CDK2 and replaces cyclin E. Once the cyclin A CDK2 complex concentration reaches a critical threshold, assembly of the pre-replication complex is terminated. The cyclin A CDK2 complex also regulates DNA replication by phosphorylating certain DNA replication machinery components. Later in the S phase, cyclin A also forms a complex with CDK1, remaining there until late in the G2 phase, when it is replaced by cyclin B. The cyclin A CDK1 complex is believed to aid in activation and stabilization of the cyclin B CDK1 complex methylene ubiquitinate. This degradation of the cyclin A CDK1 complex induces mitotic exit, in other words, the end of mitosis. Cyclin B is a mitotic cyclin which binds to CDK1 to form the maturation promoting factor, or mitosis promoting factor, abbreviated MPF. The concentration of MPF rises until mitosis, until its concentration falls abruptly due to degradation of cyclin B. High concentrations of cyclin B are necessary for cells to enter M phase, and low concentrations are needed to exit M phase. If you found this video helpful, please like and subscribe.
The cell cycle is tightly regulated to ensure proper cell growth and division. Two critical players in this process, as we saw in the video, are cyclins and cyclin-dependent kinases, or CDKs. Cyclins are specialized regulatory proteins that control the progression of the cell cycle by activating the CDKs. Cyclins themselves are synthesized and degraded in specific patterns during the cell cycle, ensuring that their activity is tightly regulated. CDKs, on the other hand, are enzymes that remain inactive until bound to a cyclin. Once activated, the cyclin-CDK complexes phosphorylate target proteins, initiating processes that drive the cell cycle forward. Together, cyclin-CDK complexes are essential for transitioning between cell cycle phases, such as from the G1 to the S phase or from the G2 to the M phase. Dysregulation of these complexes can lead to uncontrolled cell division. Just like we said before, that leads to forming tumors, which typically are cancerous tumors. And over here, you just have where the cyclins are attached and which ones are activated in each one. Cell cycle checkpoints are critical control mechanisms that ensure the fidelity of cell division. These checkpoints act as quality control systems, detecting errors and pausing the cycle to allow for corrections. G1S checkpoint. This checkpoint occurs before DNA replication begins. It checks for DNA damage, ensuring the integrity of the genetic material before it is duplicated. If damage is detected, the cell cycle is halted and repair mechanisms are activated. This checkpoint is crucial for preventing mutations from being passed to daughter cells. The G2M checkpoint. At this stage, the checkpoint ensures that DNA replication during the S phase was completed accurately and without errors. If replication errors or DNA damage are detected, the cell halts its progression to mitosis until the issues are resolved. M checkpoint. This is the spindle assembly checkpoint. This checkpoint ensures that all chromosomes are properly aligned at the metaphase plate and attached to the spindle apparatus. It prevents errors in chromosome segregation that could lead to autodiploidy. These checkpoints are vital for maintaining genomic stability, and their failure can lead to uncontrolled cell proliferation, contributing to what we're talking about the most, cancer development. So over here, I just have this little image where it gives you the checkpoints. So you have the metaphase checkpoint, the G1 checkpoint, and it tells you what it's checking for in each. Then we have the G0, the resting state, and then the G2 checkpoint, checking for cell size and DNA replication. On this slide, we're going to talk about the role of tumor suppressors. Tumor suppressor proteins are critical components of the cell cycle, acting as protective barriers against uncontrolled cell proliferation and potential tumor formation. Two key tumor suppressors are the P53, which we talked about before, and then we're going to introduce the retinoblastoma RB protein. P53 is the guardian of the genome. This protein is activated in response to DNA damage or cellular stress. P53 plays a vital role in halting the cell cycle, providing time for DNA repair mechanisms to fix the damage. If the damage is irreparable, then the P53 can initiate apoptosis or programmed cell death to prevent the propagation of defective cells. Loss or mutation of P53 is associated with many cancers, as cells without functional P53 lose their ability to effectively respond to DNA damage. The RB protein. This retinoblastoma protein is a key regulator of the G1S checkpoint. It prevents progression from the G1 phase to the S phase, inhibiting transcription factors necessary for DNA replication. This ensures that cells with damaged DNA are not allowed to replicate. Both proteins are essential for preventing the replication of damaged cells, maintaining genomic stability, and suppressing tumor development. And over here, I used this image, and it's just some tumor suppressors. Well, the P53 and where it goes and what it does and how it affects them. The dysregulation of the cell cycle has significant consequences, with the most notable being the development of cancer. This occurs when the mechanisms that ensure proper cell cycle control are compromised, leading to unchecked cellular proliferation. Uncontrolled cell division, Proper regulation of the cell cycle is essential to balance cell division and death. When this regulation is lost, cells divide uncontrollably, forming tumors that can invade surrounding tissues and spread to other parts of the body. The role of key mutations. Mutations in tumor suppressor genes like p53 and Rb, or in gene-regulating cyclins and cyclin-dependent kinases, are common in cancer. For instance, mutated p53 fails to detect and respond to DNA damage. This is going to allow the cells with genetic defects to proliferate. Similarly, manufacturing Rb proteins fail to halt the cell cycle at the G1S checkpoint. So, it bypasses the checkpoints in cancer cells. Cancer cells acquire the ability to bypass cell cycle checkpoints, enabling replication despite errors or damage. This capability earmarks cancer, highlighting the importance of innate regulatory mechanisms. Understanding these pathways provides insights into targeted therapies for cancer, aiming to restore normal cell cycle regulation. So over here, this is the image I used. This is a normal cell. This is a cancer cell. And these are cancer cells that have formed together and form a tumor. And I know that I haven't said this before the slide, but I would assume, and I hope I'm correct in this, that we know by now that not all cancers are in the form of tumors and not all tumors are cancer. Cancer treatment strategies increasingly focus on targeting the dysregulated cell cycle. A key approach involves the use of cyclin-dependent kinase inhibitors, which interfere with the aberrant cell cycle activity in cancer cells. Targeting CDKs with inhibitors. CDKs are activated by cyclins, and they are crucial for cell cycle progression. In cancer, CDKs are often overactive, driving unchecked cell proliferation. CDK inhibitors are designed to disrupt these processes, effectively halting the cancer cell cycle and reducing tumor growth. Then you have drugs like palbociclib. Palbociclib is a CDK4/6 inhibitor. It is approved for treating certain breast cancers. It specifically targets cancer cells reliant on these CDKs, inducing cell cycle arrest at the G1 phase. This precision therapy minimizes effects on normal cells, making it an effective and targeted approach. Restoring normal cell cycle control. The ultimate goal of these therapies is to reestablish proper regulation of the cell cycle, preventing cancer progression while sparing healthy tissues. Advances in this area highlight the importance of understanding cell cycle regulation to develop novel, targeted cancer treatments. And I didn't really know how to add this in there because I don't know how to cite myself. But I know that personally I had cancer, and the only way to remove it was not by drugs or therapy, but by literally removing it. So they removed all of the bad cells, so they were no longer in there to replicate to make more bad cells. And I was lucky enough that that was all that I had to do. And well, I went into remission and have been cancer-free now for 7 years. So I know that my cells were not doing what they were supposed to be doing. And sometimes you don't even feel sick. So I'm going to go ahead and use this slide to tell you that if you don't go to the doctor normally for checkups, then you should, because sometimes you don't know you're sick. And the type of cancer I had, if I would have known that I was sick, it would have been too late to do anything for me. So don't skip your doctor's visits. So to summarize what we have looked at, we have explored the intricate mechanisms that regulate the cell cycle emphasizing the essential roles of its phases, regulatory proteins, and checkpoints. So, the cell cycle is divided into distinct phases, G1, S, G2, and M, each with specific roles in cell growth and division. Key regulators, such as cyclins, CDKs, and tumor suppressor proteins like P53 and RB, ensure orderly progression through these phases. Checkpoints, such as G1S, G2M, and the M checkpoint, serve as critical quality control measures to prevent errors and maintain genomic integrity. Importance of regulation in preventing diseases. Go to the doctor. If something is wrong, then hopefully they can prevent it by regulating your cell cycle. Future directions in the cell cycle-based therapies is the advances in targeted cell cycle dysregulation, such as CDK inhibitors. They are paving the way for more effective cancer treatments. Ongoing research continues to focus on restoring normal cell cycle control and developing precision therapies tailored to individual cancer profiles. That is the end of this lesson. The last two slides are references, and I hope that everybody got something out of this and that you hopefully enjoyed it, because I enjoyed making it and I learned a few things along the way. I hope you'll have a great night.